Good morning, friends. Myself, Richa. So, today I'm going to talk about DNA replication. So, first of all, why do we need DNA replication, right? Yeah, so it is a fundamental biological process where a cell produces an identical copy of its DNA, ensuring that each new cell receives a complete set of genetic instructions during the cell division, right? So, basically, this process is semi conservative. Semi C for conservative, you can say, right? So basically, meaning what does it mean semi conservative? That each new DNA molecule contains one old one and one new strand, complementary strand, right? So here in the process of DNA replication, so many enzymes will work like DNA helicase, DNA polymerase, DNA ligase, which will work together to unwind the original DNA. So it occurs in three steps. First of all is initiation, I for initiation. Second is elongation. Third one is termination. So first one is elongation. First one is initiation, then elongation, then termination, right? So basically, here when the replication starts, that means in initiation process. So replication begins at specific sites called as ORI, that is ORI, that is origin of replication. Then what will happen in this unwinding starts, unwinding of the DNA. You can see here the DNA is getting unwinding. Here the DNA helicase will work, okay, to create a replication fork, Y shape. Replication fork is always Y shaped structure. You can see here just looking like Y shape, right? Next, then primer will work. Primase enzyme will work, right? These enzymes I will talk later. Then elongation in this DNA polymerase will work. Here you can see there are two strands, which one is leading and second one is lagging. So in this topic, I will tell you what is the difference between leading and lagging strand. In the leading strand, you can see it is a continuous. In a single piece, you can see the synthesis here. It's going on. It is towards the direction 5' to 3'. Okay. Next point here, what you have to remember in the leading strand, it requires only one RNA primer to start. It requires only one RNA primer, right? And enzymes which will work here, it's basically DNA pol, which will elongate, which helps in the elongation of the DNA. Next, you can see here, these are the lagging strand, which is also called as discontinuous. It occurs, the synthesis of the lagging strand occurs in short fragments called as Okazaki fragments. Okay, and it is always away from the replication fork. You can see here it is going away from the replication fork, but it is going from 5' into the 3' and only, right? And in this also enzymes requires like DNA pol, enzymes like DNA ligase to join the fragments together. So these are the differences which is very much important, leading and lagging strand. So the leading strand needs only one primer, right? And it is a continuous process. You can see here it is a continuous one, but the lagging strand required multiple primers. You can see here so many primers, short arrows are there, which are also called as Okazaki fragments. And later on the DNA will join together with the help of DNA ligase. So these topics like basically is frequently asked in CSN at exam, what is the difference between leading strand and what is the direction of it. So these two things, the difference and how many primers are needed there. So these things are you have to remember, right? So thank you. The next topic I will discuss about the enzymes which is required here in DNA replication. Thanks.